It should also be recording on Zoom now. So good. All right, is anyone not in yet? Cool, we'll give it another second. That's kind of cool. All right, I'm going to start it now. Straight into it. Okay. Is it? I kind of went through and edited most of mine, but hopefully. Cool. I like the music. Okay.
Okay, so if you see a question that says direct action, that's going to be ionotropic because that's the ligand, that's the faster route. And if you have metabotropic, it would be something like a GPCR or second messenger. So telling you something like indirect action. That's pretty much that one. So it's
Oh, I can add 15 seconds if we need to. Yeah. Let's just do that.
Okay. Maybe something to review. Tonic versus phasic receptors, because you guys went over senses in chapter 19 or 14, I remember which one. But you have two kinds of adaptation. You have tonic receptors, which is slow to no adaptation, and you have phasic receptors, fast adaptation. Your tonic receptors are going to have a continuous stimulus, like no adaptation with a continuous action potential. If you have a phasic receptor, the number of action potentials is going to decrease with a constant stimulus. Does that make sense? That's probably a slide you should go back and review.
And if at any point you guys do have questions, ask us before we go to the next one. How's it going? We're using what is this thing called? Quizzes. And if you guys want, you can use the code in the top left to sign in.
Okay. This might be another one you guys should review. Hair cell are the opposite of a neuron, like a normal neuron. Hair cells are weird, where actually K plus will depolarize the cell. It's just how the physics work. There's more K plus concentrated on the outside for some reason, and then it flows into the inside, and it causes depolarization that way. So that's just some hearings and stuff. Any questions?
Ooh, it's a good one.
Okay, so With the sympathetic nervous system, the answer was sympathetic nervous system dilate. So when that sympathetic nervous system is activated, the radial muscles activate and then your pupils dilate. And it's the opposite for the parasympathetic. If your parasympathetic nervous system is activated, your circular muscles will constrict, and that causes your pupils to shrink. So that's part of the vision portion. Any questions?
I think of like sympathetic fight or flight. You want to see a lot more. You want to be able to see what's around you if a lion's running at you or something. And then also, I always remembered radial as a D in it, D for dilate. It's a radial dilate. Some extra time on this one.
Okay, so this one's the opposite. If you want, so this is called accommodation. And when accommodation happens, you basically increase the curvature of your lens so you're able to see better. And this actually happens during parasympathetic stimulation, which is when your ciliary muscle contracts and your lens is rounded. If there's sympathetic stimulation, there's nothing happening, no accommodation, it's flattened. Are there any questions about that?
I also would always think about like reading a book as kind of like a calm, parasympathetic, you're not fighting anything activity. And if you're reading a book, you want to be able to see up close, you want to see those words.
Okay, this is one where you're just typing, it's a brain dump, I know what you know. Okay, that's because I put the answer in a certain way and it might have been like rated based on wordage. But could anyone tell me some features of rods? Would anyone like to tell me about rods? Anyone? Nothing? Okay. So rods.
Wait, one hand in the back.
Oh, yeah,
They're used for a good one.
yes. Anyone else want to share something about rods?
There are like a lot more rods than cones.
Yes.
Yeah.
Correct. Anyone else have anything? They contain a specific kind of opsin called rhodopsin, so those are in your rods. And then, does anyone know what color vision it produces? If it produces color vision? Rods are gray vision. Rods are black and white. Okay, does anyone know anything about cones? Nope, okay. Cones have cone opsin, so that's one way you can distinguish between the two. Road opsin, rod, cone opsin, cones. And cones respond to color wavelengths, so color vision. So how I think about cones is cone, CO, color, CO. Just letter association. And there are less of those and they're less sensitive to light. Any questions about that?
Does anyone know where you would find cones in the eye?
In the phobia,
In the fovea? And is that like in the center or the periphery?
like,
The center.
Center. Yeah. There's the macula that contains the fovea, and that's like the very condensed region with a ton of cones kind of in the back center of your eye. Cool. Any other eye questions?
Okay,
Nice.
so does anyone want to tell us what the answer is to that? Anyone who got it right want to share with the class? Okay, it's mechanical and thermal nociceptors. So the fast pain pathways, like the sharp tinge you feel when maybe you put your arm on something, or if you feel a burn, and that's mechanical and thermal nociceptors. Olfactory and gustatory are your sense of smell and taste. Any questions?
Nice.
Okay, so olfaction and gustation always use metabotropic. That's like one that I was able to make an association with in my head. Even if I didn't remember the pathway or the mechanism, I was always like, those two are metabotropic. They're always going to be a longer pathway. Yeah.
Yeah. See ya. What is that guard?
What?
You let that happen once.
I don't like this. Okay,
Okay.
so cholinergic receptors bind acetylcholine. Adrenergic receptors bind epi/norepi. Acetylcholine, cholinergic, choline, kind of match that together. Adrenergic, epi and norepi are secreted by the adrenal gland.
Cool. Mhm.
Okay, this is another open response one.
Pretty sunset. It's a good sensitive there. Oh, that's the clock in the took me way too long to figure that out.
Okay. Again, ignore this. Does anyone want to tell me anything about the somatosensory cortex? Okay, so it processes skin-based sensory information. It includes thermoreceptors, nociceptors, mechanoreceptors, and it's located in the postcentral gyrus. The primary motor cortex is voluntary skeletal muscle control, and it involves speech production, elaboration of thoughts as well, and it's also in the precentral gyrus. Okay, the answer was the cerebellum for that one. Are there any questions about that? Okay. Like,
Some more time.
okay, so the hypothalamus, think homeostasis. So when you guys got to your endocrine unit, you're basically focusing on the hypothalamus and hormones. And that's kind of like always hormones, homeostasis, hypothalamus, homeostasis. That's an association I made. So everything except for monitoring the digestive organs in the heart were part of the hypothalamus function. Monitoring the digestive organs and heart is part of your brain stem. It is one of the more basic functions of your brain, which originated in the brain stem, which was developed first. Okay, so white matter, myelinated axons, and then cell bodies and interneurons are in your gray matter. Okay, then ignore the score. Okay, so first in your reflex are a sensory receptor response to a stimulus. Then the afferent pathway relays the stimulus away from where the receptor was towards your integrating center, which is your spine and your brain. Then your integrating center processes the available information and then sends it through the efferent pathway. Once it goes through the efferent pathway, it goes to the effector, which could be one of your organs, your muscles, whatever, and then they carry out the response. So that's just like the basic steps of a reflex arc. Okay, so does anyone want to tell me what postganglionic fibers release?
They see the answer. Yeah.
Oh, okay. Okay, yeah, that's the answer. Cool.
Does anyone have any questions about that stuff? Cool. I'm gonna add some time.
Okay,
Nice.
so yeah, that was the answer. There is a question about it.
Did he say in class whether he wants you to know all of these letters? He said he doesn't want you to know them. Okay, then kind of ignore this one, but I'll still talk about it briefly. And also ignore 1 and 2 from now because it's also kind of like this. But um, the A band is simply the myosin, and so the myosin itself, the length of it is never going to shorten. The I band is talking about the like actin filaments that are going to be shortened because they're pulled in the sliding filament. Um, don't worry about the rest of it. A good way to memorize it is that the A band is always the same. So there's that. Yep, nice. Anyone want to say what is false about this? If not. So that's talking about the actual cross bridge cycle. Once we start doing the whole actin and myosin movement, this is a little bit before that. Lily, you had your hand up?
Is it that it binds to the troponin?
Yep, you got it. So again, I did this on the last one. I'll do it again here. Very often he likes to do a true or false where most of it is true, but maybe one word is what's false. And he'll want you to cross that out, put the right thing. So this is true following an action potential at the NMJ. An influx of calcium does occur, but it binds to troponin, and troponin is what is kind of connected to the tropomyosin and pulls that off the actin, allowing for the whole cross bridge to go. Cool. Again, here's another one where I'm using the AI band, whatever that stuff. If you know it, that's good. But yep, Z disk. You can define a sarcomere as the boundary between Z disk to Z disk. Give extra job for this one. Nice. Basically, everyone got that one right. So yeah, it's the this is like step one of the power stroke is after it's done the whole thing, we're back to the beginning and we still have the myosin bound to actin and you need the ATP, a fresh new source of ATP to bind for it to be able to detach. And so basically when someone's dead, they can't be making ATP, so they don't have enough. And so those muscles stay locked for a long time. Yeah, so G actin is like every individual ball of actin in that actin filament. F actin stands for filamentous actin. So in the cycle in your textbook, I would recommend memorizing that exact photo of that cycle. But it says specifically we're having myosin bind to the G actin units. Okay. This one, straight from your textbook. I know it's very kind of like niche thing, but troponin T. Well, first off, your textbook describes troponin kind of like a golf club where you have like a long handle and then the club component of it. Troponin T is, think T tropomyosin. That's the handle that's binding to the long sheath of tropomyosin. Troponin I is the answer here. That's the part that binds to actin. That's like the golf club of the troponin. And then TNC, troponin C, C stands for calcium. That's the part that binds to calcium. And then Z is not a thing. Yeah.
Wait, what was TNT again?
TNT is the part of actin that binds to tropomyosin. So it's like the handle of a golf club. Yep. It's not bad. So I'll go through one by one because everything else that isn't the right answer is something you should know. So the first one here, um, a protein that changes conformation to open ryanodine receptors. Does anyone know what that is? I think you said it. D. Yeah, the dihydropyridine receptors, the DH PRs or whatever it is. That's what that is. An area within the muscle cell that releases heavy amounts of calcium ions after excitation. What is that? Yep, sarcoplasmic reticulum. An enzyme that breaks down acetylcholine within the NMJ. What is that? Acetylcholine esterase. And then, yeah, the T tubule is a part of the sarcolemma that basically dips down so you can have the action potential spread all the way down, leading to the excitation of the muscle. You kind of have one like this, but it's okay. Yeah, use extra practice. Yeah, so this one's false. Again, there's one word here that is, there's a little component here that's false. Um, so acetylcholine does bind ligand-gated channels. Does anyone know what type of channel is at the NMJ? Starts with an N. Nicotinic. Yeah, whoever said that. That's what opens. But those don't create the action potential right there. That creates a graded potential that then kind of travels to a different part of the membrane. That's where the action potential does take off. So if you need to review that, I think that's like chapter 13 in your books. Maybe go review a little bit more about like motor end plate and all that stuff. Everyone feel good about that? Nice. You guys got this. Let me get the. I had a photo that was supposed to come up here. Here we go. This is this part of your book where you don't want it to be too much overlap between the actin and myosin, and you don't want too little. There's an optimal amount where you get perfect amounts of cross bridge formation. So yeah, the first one, that is true. Smooth muscle is not striated, and it can be characterized by the number of gap junctions. Does anyone know what two ways we can describe smooth muscle is based on that? Like what the terms are? Yeah. Single unit, multi-unit. Yeah, which one would have more gap junctions? Single unit. Yeah. It's one unit, so we want a lot of metabolic coupling, and that's what we use those gap junctions for. Next thing, unlike skeletal muscle, smooth muscle is uninucleated. That's true. This one's the false one. So it says the active MLCK activates calmodulin, which phosphorylates the myosin head. That's flipped. So calmodulin, once it's bound to calcium, then goes and activates MLCK. And MLCK stands for myosin light chain kinase. Anyone want to say what a kinase does? Yeah, it's phosphorylates. So kinases add phosphates. And so that's what's going to do it to the myosin head, and that's what we need for smooth muscle. And then the last thing, yep, the autonomic nervous system does innervate smooth muscle. We get more time. Okay. So there's three ways we characterize hormones. There's steroid hormones, peptide hormones, and amine hormones. Estrogen is a steroid hormone. ADH is a peptide hormone. It's released by the pituitary. Specifically, which side of the pituitary is ADH released from? Anterior or posterior? Posterior. So ADH is the same as vasopressin. That's one of those two posterior pituitary hormones. And that one is a peptide hormone. Aldosterone is not a peptide hormone, though. Does anyone want to guess what aldosterone is? What type? Steroid. Yep. And you'll notice that most steroid hormones have like ster in their name. Aldosterone. So kind of a good indicator there. And then amine hormones, those are your epi and norepi. So um, cool. Anyone have any questions about this? Hormone unit is really like you either memorize it or you don't. Like flashcards or whiteboards, probably the way to go. Yeah. Cool. So peptide hormones, are they hydrophilic or hydrophobic? Hydrophilic. Yep. Steroid hormones are hydrophobic. And so steroid hormones, since they're hydrophobic, can go straight through the membrane and enter in and do a lot of crazy stuff to the cell. Oftentimes they're going to go to the nucleus and then become a transcription factor or kind of dimerize to become a transcription factor. Whereas peptide hormones, they can't make their way easily through the membrane because they're hydrophilic. So they bind to what kind of protein most often? What's a membrane protein we've talked about for a long time now? Not transverse, GPCR. A lot of times peptide hormones bind to GPCRs, and that's going to have a more immediate and transient, meaning like shorter lasting effect than steroid hormones. And again, that's because steroid hormones go into the cell and do affect transcription and like all the DNA central dogma stuff. Everyone feel okay about that stuff? There you go. Helped you all out in this one. How's it going? Hello. So, yep, ADH is posterior pituitary. Let's see, where do I have the little thing? Um, this photo right here, you can't see it that well. Anyone who's taken the MCAT is going to. Flat peg is like one of the best mnemonics you can use for the MCAT. That stands for all of the anterior pituitary hormones. Sorry, it's really small, but you have the F for FSH, L for LH, A for ACTH, T for TSH, P for prolactin, E, it releases endorphins. You don't need to know that part as much. And then G for growth hormone. So this is a really good mnemonic for all those anterior pituitary. Nice. Okay. So again, um, this is kind of the pituitary gland. We have the hypothalamus up here doing its thing. This is the anterior. This is the posterior. And the big difference is that, first off, what hormones are going to be released by them? There's a certain set that are anterior, some that are posterior. But the anterior pituitary uses a portal system. Does anyone know what a portal system is? If it's okay if you don't. That one's kind of a weird term. It's when you have a set of capillary beds, two of them back to back. There's very few portal systems in the body. There's one like in the liver. But basically, that means that we are not talking about neural tissue when we're talking about the anterior pituitary. That is going to be something connected by vessels, but the posterior pituitary is an extension of axons basically from the hypothalamus. So the posterior pituitary is neural tissue. Everyone feel good about that? All right. So yeah, definitely memorize every one of these hormone axes. I think honestly the slides were better than the textbook at showing them. So I would probably look at the photos in the slides. Um, Anyone want to tell me what's wrong with this one down here? Very small detail, but yep. Um, It's not that, but it's the order of the hormones. So the hypothalamus releases PRH, and then the anterior pituitary is the one that releases prolactin, so the PRL. Most of the time, the like releasing hormone is coming from the hypothalamus, going to the anterior pituitary, and that's where the next hormone goes through. But yeah, these other ones are right. TRH to TSH to make thyroid hormone T3 and T4. Um, CRH goes to make ACTH, that makes cortisol. It's also important that you memorize like what organ is going to affect, whether it's like the adrenal gland, the liver, the thyroid, that sort of thing. And then yeah, GH, RH, growth hormone, releasing hormone, stimulates growth hormone, and that makes IGF-1 in the liver. So for the stress response, I always thought this was kind of more intuitive. Like if you're being, chased by a bear, there's certain things your body you would want it to do, certain things you wouldn't want it to do. Inhibiting energy storage, you wouldn't want to store your energy, right? You want to get rid of it, you want to release it. So that is true. We do inhibit energy storage. We enhance cardiovascular function. We want to be like, heart beating. We want to be breathing well. We don't care about the kidney though. We wouldn't like pull over to go pee. So kidney, don't worry about that. And um, but we are also like inhibiting reproduction, that sort of thing. Yep, false. Um, Anyone want to say what's false about this statement? Yep.
Does it increase the number of?
Yep. Does anyone remember the like specific name of the aquaporin that goes in the collecting duct? Super niche, but you have AQ2, so you have the aquaporin 2 in the collecting duct, and that's going to pull water out of your like kidney system basically, and then it's going to exit through AQ3. So you have aquaporin 3 that's going to dump the water back into your bloodstream basically. That's about the maximum level of detail he cares about for now. You'll get into the kidneys in a little bit, but I think, yeah, again, you can't really see that photo super well, but this is what I'm talking about here. Um, yeah, that's the last question we have on this one. So, end assessment, 43% average, not bad, not bad.